Hello dear students, good day to you all. I am Dr. Geetanjali Muralidharan. I'll be taking the microbiology classes for you. So today being the first class, we'll have an introduction to the subject and we'll see some important aspects of history of the specialty, that is the pioneers who laid the foundation for our subject. And then we'll move on to the topic morphology of bacteria. So all the important aspects, all the topics we'll cover in a way that we will deal with the important questions which were asked in the previous year university exams. So I'd like to modify the name of our subject as clinical microbiology. Why? Because you all know microbiology is the study of microorganisms. That means living organisms which cannot be seen by our naked eye, and you need microscopes to visualize them. So we know there are a number of microorganisms in the world, millions of microbes. We don't need to learn about all of them. So we'll be focusing only on the microbes which are pathogenic to human beings. Pathogenic to human beings means that which can cause infections in humans. So we'll be dealing with only the microbes which are pathogenic. So we can call our subject as clinical microbiology because we are learning the microbes which cause infections in humans, or you can call the subject as infectious diseases. So whatever specialty we are going to choose in medical or dental, any field, any medical field, you'll have to deal with infections, isn't it? So we have to definitely learn about what are the causative agents of various infections. And then we'll see the pioneers in the specialty and a few landmark discoveries in microbiology. So when you hear microbes, the thing which will come to your mind may be the ads which we see in television, right? Toothpaste ads, that is dental problems, toothache or abdominal pain, and the cause is bacteria. That is what they highlight. And that, so we have this image in our mind when we think about microbes, right? There are various microbes, not just bacteria. The one which we commonly hear is bacteria. Now we are very familiar with viruses. We are hearing new and new viruses each day that is causing nightmare to us. But all these things come under microbes. So we have to give a kingdom to these organisms. You know, it's not plant kingdom, it's not animal kingdom. We have to assign a kingdom for microorganisms. So for that, we have chosen kingdom Protista. So that is a kingdom in which microorganisms belong to. And microorganisms can be either prokaryotes or eukaryotes, because we know bacteria, they belong to prokaryotes, and all other microbes except viruses belong to a separate entity which we'll deal later. It's actually a transition between living and non-living. And all other microbes, that is the ones which you have already learned in your school days, like fungi, parasites, all of them are eukaryotes. Only bacteria come under prokaryotes. So being familiar with all these terms, that is, we have bacteria, then we have viruses, fungi, and parasites. Right? So these are the different microbes which we know. So we can divide the microbiology specialty into various branches based on the organism which we are dealing with. So when we are dealing with bacteria, you can call the specialty as bacteriology, and when it is virus, you can call it as virology, and if it is parasites, you can call it as parasitology. And for fungus, we have a different term, that is mycology, because fungal infections is called as mycotic infections. So you can call the specialty as mycology. In addition to all these things, all these branches, you have another very vast branch of microbiology called as immunology, where you learn about the immunological aspects of diseases. So it's a very interesting field. So that's also a branch of microbiology. So coming to the history of microbiology, we'll see a few persons. Some of the faces may be familiar to you. You might have seen in your +2 textbooks. So the first person who discovered microscope, he's Anton van Leeuwenhoek. Okay. Now the second person, that was in 1600s. You remember, you just think of the time when he discovered microscope. The first microscope was in 1600s. So then the next person, who is a very important person, you can see he's giving some injection to a boy. So this person is Jenner. So when you hear the name Jenner, you might be remembering what was his discovery. He discovered vaccine. And the first vaccine which he discovered was smallpox vaccine. Okay, so that was the basic foundation for development of all other vaccines. So that was what led to eradication of smallpox vaccine. So that was by Edward Jenner. Now the third person, he's a famous person now, but at his time, whatever ideas he propagated, it was not accepted by the scientific community. And that idea which he proposed at that time, that we take for granted now, hand washing or hand rub, hand hygiene. So he was the first person who said you should wash your hands. Doctors and other medical persons should wash your hands to prevent diseases in their patients. At that time, nobody know infection. They don't know the term infection. They don't know it is caused by microbes. But he proposed the idea of hand washing, and his name is Ignaz Semmelweis. So he faced a lot of criticism for proposing hand washing idea, and that time he was not considered by any of the scientific people, but now we take his idea for granted. So he's the father of infection control practices, Ignaz Semmelweis. Okay, so now we'll see the pioneers of microbiology who laid the foundation for this subject. So some of these pictures may be familiar to you. The first picture, that is the person who discovered microscopes. So you might have learned in your +2 classes. This is Anton van Leeuwenhoek, and that was in 1600s, long back. So he's the father of microscopy, the person who discovered the first microscope. And so the next person, you can see he's giving some injection to a small boy. So this is the person who first discovered vaccine. So now we know a lot of vaccines. It was a time when there was no vaccine available. People don't even know infections is there. It's caused by microbes. So at that time, this was Edward Jenner. So you may be remembering the name Jenner. That is the person who discovered the first vaccine, and the vaccine he discovered was smallpox vaccine. So now this disease doesn't exist, right? Smallpox is eradicated. This was because of a successful vaccine developed against smallpox. So Edward Jenner, that was in 1796, 1700s. That time Jenner discovered the first vaccine, and that vaccine was for smallpox. Now the third person, he's a very important person. That is, he introduced the idea of hand washing. In a time when people don't know about infections, he found that his patients were dying after surgery, after delivery. Lot of mothers were dying. So he told that people who work in this medical field should wash their hands so that they can control diseases in their patients. They can reduce the diseases in their patients, reduce the mortality, death rate can be reduced. But nobody believed him, and he was not accepted by the scientific community. But now we take his idea for granted. So that is hand hygiene or hand washing. So that idea was proposed by Ignaz Semmelweis. So please remember his name, Semmelweis. He is the father of infection control practices, the person who first introduced hand washing. Okay, now with these people, so please remember Anton van Leeuwenhoek, who discovered microscope, Edward Jenner, vaccine, and Ignaz Semmelweis, who introduced hand washing. Now we move on to the most important personalities of microbiology, which you all may be knowing. The first person who's called as father of microbiology, and the second person he's called as father of bacteriology or modern microbiology. So who is the first person? The person who introduced germ theory, the one who said infection is caused by microorganisms, and microorganisms do not arise spontaneously. They will arise only from their own kind. That is the idea of germ theory. He disproved the idea of spontaneous generation. So is none other than Louis Pasteur. So this is a favorite question of all the examiners. Louis Pasteur, father of microbiology. So that you can't forget. When you learn the subject microbiology, you should never forget Pasteur. So don't forget Louis Pasteur, for that is a favorite question. Even for MCQs, for a neat question, father of microbiology, who's the father of microbiology is always asked, and don't forget his name, Louis Pasteur. So what about the other person? That is Robert Koch. So both of them were actually of the same ages, like at the same time, 1800s. Both of them had a lot of discoveries. They did a lot of discoveries in the same time. And it was Koch who proved the theory of Pasteur. Pasteur said It is germ theory, that is germs are responsible for causing infection, and Koch was able to prove the theory of germ theory, and he laid many other discoveries, like he introduced the staining techniques, he introduced solid culture media, he discovered many important bacteria, and so both of them are the most important personalities, Louis Pasteur being father of microbiology and Robert Koch being father of bacteriology or modern microbiology. And Koch was awarded Nobel Prize for discovery of a very famous bacteria which causes tuberculosis, that is Mycobacterium tuberculosis. So why these two persons are very important for us? Because for exam they always ask, so these are the previous year questions: contributions of Louis Pasteur, contributions of Robert Koch, and what are Koch postulates. So these three are the questions that you can get from history part. Okay, so we will see the points, what are the contributions of Louis Pasteur for exam purpose. So we'll see what are the contributions of Louis Pasteur. So we know that is he's known as father of microbiology. We don't forget that. You don't forget that. Okay, that's very important. Then why he's called as father of microbiology? Because he's the person who disproved spontaneous generation theory. You may be remembering the Swan-Neck experiments. You might have learned in your school classes. With the Swan-Neck experiments, Pasteur discovered that, or Pasteur realized that microbes do not generate spontaneously, they generate only from their own kind, and microbes are responsible for causing spoilage of food, spoilage of growth, and that's why it may be the reason for infections also, causing diseases in people also. So he postulated germ theory and he disproved spontaneous generation theory. Germ theory means germs causes infections. So that is the most important discovery, most important concept that was proposed by Pasteur. Then he introduced the concepts of fermentation. Microbes causes fermentation, fermentation of food and other edible things. So that is another thing that Pasteur proposed, that is concept of fermentation. Then many concepts of sterilization, because he realized that heating food can increase the shelf life of food. So he introduced pasteurization, that is he heated milk, and milk can be retained for more days. When milk is heated, the microbes are destroyed. So that concept was proposed by Pasteur. He did some experiments and realized heating is an important method of killing microbes. So thus he discovered pasteurization. Then other sterilization methods like hot air oven, steam sterilization, that is your autoclave, all these concepts were proposed by Pasteur. Then the idea of attenuated vaccines. Attenuated vaccine means when microbes are weakened, the live microbes, when they are weakened, they lose the ability to cause disease, but they can be used to prevent disease. When you inject them, the people become immune to the disease. So that idea was also introduced by Pasteur. And you should remember what are the vaccines which were discovered by Pasteur: chicken cholera, anthrax, and rabies. So we'll give a code for that: CAR. Okay, cholera, anthrax, rabies. So these vaccines were discovered by Louis Pasteur, and he introduced the concept of liquid media. He used nutrient broth to grow microbes. He realized that microbes can grow in broth when sufficient nutrition is provided. So the idea of liquid media, and he found the Pasteur Institute in Paris. So he's the founder of Pasteur Institute. So we can remember a few points, like at least 4 points you should remember: proposed germ theory, disproved spontaneous generation theory, concept of fermentation, then pasteurization, steam sterilization, and hot air oven, all are concepts of sterilization. Then he discovered the vaccines: cholera, anthrax, and rabies. So these are the contributions of Louis Pasteur, which you should remember for exams, when you write for exam. Okay, so now we have seen the contributions of Pasteur. So we should know what are the contributions of Robert Koch. So I told you Robert Koch is called as father of bacteriology. Why? Because most of his experiments were on bacteria. He tried to develop more and more methods of cultivating bacteria, demonstrating bacteria under microscope by using staining methods. And his discoveries, his experiments helped to prove the germ theory which was laid by Pasteur. So see, he has discovered anthrax bacilli, TB bacilli, and cholera bacilli. So I think here we can give a code as CAT. before we set CAR as the code for the vaccines which were discovered by Pasteur. So here discoveries of Robert Koch, cholera bacilli, anthrax bacilli and TB bacilli. So among this, the most important discovery for which he received Nobel Prize, that is TB bacilli. So the bacteria which causes tuberculosis, it's called as Mycobacterium tuberculosis. So all these discoveries were done by Koch, and he developed staining techniques for bacteria, like he used simple stains which give more contrast to the bacteria and was more easily visible under the microscopes. So that was also introduced by Koch, and he first isolated bacteria in pure culture. So I told you Pasteur introduced the uh, liquid media for cultivating bacteria. He found that bacteria could grow in liquid culture media. Solid culture media was first introduced by Robert Koch, so that he was able to, in solid media, he was able to grow bacteria as bacterial colonies. So that was first done by Robert Koch. And Koch postulates, among all these things, Koch postulates is more important in exam point of view. So what is Koch postulates? So Koch postulates was actually proposed in relation to anthrax bacilli. While Koch was studying about anthrax bacilli, what he noticed, all those things he put under as postulates, and there are four postulates that is called as Koch postulates. So what he said first is, so suppose we can say it with the example of an anthrax bacilli. So anthrax bacilli should always be associated with that disease anthrax. Anthrax will produce certain skin lesions. It will lead to death of the animal. So whichever animal is having this disease, this organism should always be constantly associated with this particular disease. Now the second postulate is, when you take the lesions from that animal and you try to grow this bacteria in lab, in culture media, you put the lesion samples into the culture media, you should be able to grow the organism in culture media. So microorganisms should be isolated from the samples which is obtained from the diseased animal. So what is third postulate? So now we have grown the organism from the sample which is obtained from a diseased animal. You have grown the organism in culture. Suppose you are inoculating this culture into another healthy animal, that animal should also develop disease. So see, this is second animal. When you introduce this cultures into this animal, that animal also develop disease. So that is the third postulate. Again, you should be able to re-isolate the organism from this second animal. So first animal has got the disease, and microorganism should be associated with this disease. And when you take the sample from the first animal and grow it in culture, try to inoculate it into liquid media and solid media, you should be able to grow the microorganism in lab. Now this cultures you are inoculating to a second healthy animal, this animal should also develop the same type of disease. Again, when you take the sample from the second animal and grow it in lab, in culture media, you should be able to grow the same organism. So all these things are interlinked. These four are the Koch postulates. So that is the most important question in exam point of view, and all other contributions of Koch also you should remember. Please remember the three bacteria which he discovered: cholera, anthrax and TB bacilli. So that is enough for history, and one more person I will mention, that is Lister, which I have not shown an image. Joseph Lister, he is considered as father of antiseptic surgery because he is the first person who introduced the use of antiseptic to prevent wound infections. Okay, so that is another person. Please remember his name also. But what is more important is Pasteur and Koch. Both of their contributions is more important in exam point of view. So with this, we wind up this class, and we'll continue with the morphology of bacteria in the next class. Thank you.